Yeah, I think it's working. Yeah, today, the last day, but still we have a full day to go of interesting talks to look forward to. And so it's my pleasure to introduce our third plenary lecture by Jason Schweinsberg. He's a professor in the math department at UCSD, and he's did his PhD in Berkeley, did his postdoc in Cornell, and he's been working on stochastic processes, and lately has been applying some of this stuff to cancer. Well, there you go.
Sorry, could you put on the microphone?
Thanks. Okay. Thank you, and thanks to the organizers for the invitation to speak here. So as we've heard in several of the talks earlier this week, especially on Wednesday, genetic data can be very useful for studying the evolution of cancer. And in this talk, I'm going to discuss this issue from a mathematical perspective and present some theoretical results on the site frequency spectrum for samples taken from growing populations. So I'll begin by giving a brief overview of coalescent theory, and I'll define the site frequency spectrum. Then I'll review some results for the site frequency spectrum in the standard coalescent model, which is Kingman's coalescent. Next, I'll present some results for an exponentially growing population. This part of the talk is based on joint work with Kit Curtius, Brian Johnson, and Yubo Shuai. I'll present the theory behind some of the work that Kit Curtius presented on Wednesday, and I'll also present some additional follow-up joint work with Yubo. And then in the last part of the talk, I will discuss some results for a population model in which spatial constraints prevent the population from growing exponentially fast. And this part of the talk is based on joint work with Yubo and with Shirshindu Ganguly of UC Berkeley. So I'll begin with a brief overview of coalescent theory, which has been an essential tool in population genetics since the seminal work of John Kingman in 1982. Kingman demonstrated that sometimes, instead of studying the evolution of a population going forwards in time, it's useful to think about what happens when we trace ancestral lines backwards in time. So let's suppose we have a sample of lowercase n individuals from a population at some time, and we imagine following the ancestral lines of these individuals backwards in time. And then these lineages will coalesce until eventually we trace the entire sample back to one common ancestor. And so here's a picture involving a sample of size 6. We have these 6 individuals that we sampled from the population. And as we trace back the ancestral lines, first lineages 1 and 2 merge, which means that these 2 individuals have a common ancestor at this time here. Then individuals 4 and 5 merge. Then 3 merges with 1 and 2. 6 merges with 4 and 5. And finally, the last 2 lineages merge. And at that point, we've traced the entire sample back to one common ancestor. The resulting tree that we get is sometimes called a coalescent tree, and it depicts the ancestral relationships among the sampled individuals. Now, when we take a sample of individuals from a population, we can sequence their DNA. And that allows us to see that mutations have appeared on certain subsets of the sampled individuals. And it's often useful to summarize this information by considering what's known as the site frequency spectrum. So we're going to denote by MKN the number of mutations that are inherited by exactly k out of the n sampled individuals. And then these numbers MKN, as k ranges from 1 to n minus 1, are called the site frequency spectrum. So in this picture, there are now dots indicating the times of mutations, and 3 of the mutations are inherited by only 1 of the sampled individuals. This mutation is inherited by individual 1, this one is inherited by individual 2, and this one is inherited by individual 6, so M13 is equal to 1. This mutation here is inherited by the individuals 4 and 5, so M2n is equal to 1. And these 2 mutations at the top are each inherited by 3 of the sampled individuals, so M3n is equal to 2. Now, the site frequency spectrum is closely related to the branch lengths in the coalescent tree. So external branches are branches of the tree that lead directly to a leaf. Those are the blue branches in the picture. And I'm going to denote by Ln with the superscript Ex the sum of the lengths of all of the external branches. The other branches are said to be internal, and I'll denote by Ln with the superscript In the sum of the lengths of all of the internal branches. I'll also denote by Lnk the sum of the lengths of all of the branches that support k leaves, which means that they lie along the ancestral line of k of the n sampled individuals. So in the picture, the blue external branches each support one leaf. These two orange branches each support two leaves, because this orange branch lies along the ancestral line of the individuals 1 and 2, and this orange branch lies along the ancestral line of the individuals 4 and 5. And the two red branches each support 3 leaves. This red branch is on the ancestral line of the individuals 1, 2, and 3, and this red branch is ancestral to 4, 5, and 6. Now, notice that if a mutation appears on a branch that supports k leaves, then that mutation will be inherited by k of the n sampled individuals. And therefore, if we assume that mutations appear at the constant rate nu along each lineage, then the conditional distribution of mkn given lkn is simply Poisson with a mean of nu times lkn. And so the point is that if we understand the distribution of the lkn's, we automatically understand the distribution of the mkn's. And so for most of the rest of the talk, I'm going to focus almost entirely on understanding the branch lengths, the lkn's. So next, I want to review some results for the standard coalescent model, which is known as Kingman's coalescent. I'll begin by introducing the classical Morin population model, which goes back to 1958. In the Morin model, the population size is fixed, so there are exactly 2n individuals in the population at all times. Each individual independently lives for an exponentially distributed time with rate 1, and whenever an individual dies, a new individual is born, and we choose an individual at random from the population to be the parent of the new individual that's born. So in this model, suppose we choose 2 individuals at random from the population at some time, and we let capital T be the amount of time that we have to look back to find a common ancestor of these 2 individuals. And then in this case, if we take T divided by n, this quantity has exactly an exponential distribution with rate 1. And from that result, we can see that the genealogy of the population in the Morin model can be described by a process known as Kingman's coalescent, which goes back to the original 1982 paper of Kingman. And Kingman's coalescent has the property that when we are tracing back ancestral lines, each pair of lineages merges at rate 1. And so whenever we are down to k lineages, there are k choose 2 pairs of lineages that could merge, each of them merging at rate 1, so the total merger rate is k choose 2. So this means that the time until the next merger is exponentially distributed with a rate of k choose 2, and then after that exponentially distributed waiting time, we pick 2 lineages at random and merge them together. And Kingman's coalescent is by far the most widely studied coalescent model, because it not only describes the genealogy of the population in the Morin model, but it describes the genealogy of populations under a wide range of circumstances, as long as the size of the population is nearly constant over time. So we can consider some basic results for the site frequency spectrum of Kingman's coalescent. In 1995, Fu showed that the expected value of the total length of all branches that support k leaves is exactly equal to 2 over k. And therefore, if mutations appear at the rate nu along each lineage, the expected value of mkn is 2 times nu over k. If we take k equal to 1, we see that the expected value of the total external branch length is exactly equal to 2. And to compute the expected value of the total internal branch length, we sum 2 over k over k from 2 to n minus 1, and this expression is approximately 2 log n. Now, one can also consider the fluctuations around the mean. And in 2011, Janssen and Kirsting showed that the total external branch length, its distribution is asymptotically normal. That is, if we take the total external branch length, subtract the mean of 2, and we standardize by multiplying by the square root of n over 4 log n, this converges in distribution to the standard normal distribution. And then in 2015, Dahmer and Kirsting obtained a similar result for the internal branch lengths. We'll let mu k be 2 over k. That's the expected value of LKN. And then for any positive integer k, we can consider the joint distribution of the branch lengths that support between 1 and capital K leaves. We'll standardize by subtracting the mean from each coordinate, and we'll multiply again by the square root of n over 4 log n. And Dahmer and Kirsting showed that these random vectors converge as n goes to infinity to a multivariate normal distribution where the coordinates are independent. This covariance matrix here is the k by k identity matrix. So for Kingman's coalescent, everything is well understood. However, for the populations that we're most interested in studying, they will not have nearly constant size. We're interested in studying cancer, and cancerous tumors tend to grow rapidly over time. And therefore, Kingman's coalescent is not a suitable model for the genealogy of the population. So in the next part of the talk, I'm going to discuss modeling tumor growth by using a birth and death process. So we begin with one individual at time 0, which we think of as being a cell that has just acquired a cancer-causing mutation. And then each individual independently gives birth at rate lambda, and each individual independently dies after an exponentially distributed time with rate mu. We will assume that lambda is greater than mu, so that we're in the supercritical case, and we'll let r be equal to lambda minus mu. So let's let x of t be the population size at time t, and then the expected value of x of t is e to the rt. So the population size is growing exponentially over time, and this parameter r indicates the exponential growth rate. Now, more precisely, if we take e to the minus r times t times x of t, then this converges as t goes to infinity to some limiting random variable w. And conditional on the event that the population doesn't go extinct, the random variable w has an exponential distribution. So now we're going to take a sample of n cells from this population after some large time capital T, and we're interested in understanding the genealogy of this sample of size n, and more precisely, to understand what we would expect to see in the site frequency spectrum. Now, when a population grows exponentially fast, the genealogical tree looks quite different from what we see in Kingman's coalescent when the size of the population is fixed. And the reason for this is that when the population is growing exponentially, then after some relatively short time t, the population may already be very large. And furthermore, many of the individuals alive at time little t are likely to have descendants still alive in the population after some much larger time capital T. And so if we take a sample from the population after this very large time capital T, then it is very likely that all of those sampled individuals are going to be descended from distinct ancestors at the much earlier time little t. And as a result, the genealogical tree tends to be star-shaped, as in the picture on the left.
Topologically, they're really the same, aren't they? So the word shape, I'm confused. It's really more a question of the sizes.
Okay, that's right. If you're just concerned with the topological shape of the tree, then yes, they are identical in these two models. But what's different are the branch lengths.
Yeah.
In the case of the tree on the left, we have what's called a star-shaped tree, where we have these very long external branches, and most of the coalescence occurs near the top of the tree.
Is there a definition of star-shaped?
I mean, strictly speaking, the definition of star-shaped would be that all of the lineages coalesce exactly at one point at the root. So this would be, I would say, approximately a star-shaped tree.
Another difference will be the surviving leaves. In the constant population, you might have a lot of mutation get lost around the gas and
well, that's reflected here in the shape of the tree, I would say. Yeah. Okay, now there has already been quite a bit of earlier work on the site frequency spectrum for supercritical birth and death processes. In 2013, Rick Durrett showed that if we fix a number k between 2 and n-1, then as the time t at which we take the sample tends to infinity, the expected number of mutations that appear on k of the n sampled individuals converges to the sample size n times the mutation rate nu divided by the growth rate r times 1 over k times k minus 1. The expected number of mutations that appear on only one of the sampled individuals tends to infinity as t goes to infinity, and it's asymptotically equivalent to n times nu times t. And as we saw in Kit Curtius's talk on Wednesday, this 1 over k times k minus 1 pattern fits very well certain data on blood cancer. Now, similar results were derived and applied to cancer data in these 3 papers here, which came out a little bit later. I also want to mention that Gunnarsson, Leader, and Fu calculated the exact expected value of the site frequency spectrum in the case when we have data on the entire population rather than just a sample from the population. And Gunnarsson, Leader, and Zhang obtained a law of large numbers in that context. So our primary goal here is to obtain sharper asymptotic results for the site frequency spectrum. We would like to understand not just the expected value, but also something about the distribution of these quantities. The idea being that if we understand the distribution of these quantities, then we can use those results for statistical inference, as we saw in Kit Curtius's talk on Wednesday. Now, to understand to obtain these sharper results for the site frequency spectrum, we're going to want to think about the genealogical trees in a little bit different way. And so I want to start by doing this in the simple case of a Yule process where there are no deaths and each individual gives birth at rate r. So we can construct a Yule process up to time t in the following non-standard way. So we begin with one individual at time 0, and because this individual can never die, we're just going to draw a line representing this individual all the way down to time t. So now we have to find the branch points, the times when this individual gives birth. But rather than starting at time 0, I'm going to start at time t and look backwards along this lineage until we find a branch point. And maybe there's one about here. So a new individual is born at this time, and I'll draw a black line representing that individual down to time t. And then I'm going to color this segment red, because we've already seen that there are no branch points in that segment. But we haven't yet looked for branch points along the black line. So we'll do that next, again, starting from time t. And maybe there's a branch point here. So we draw another line down to time t, and we color this segment red. And then we repeat this process. Maybe next time we find no branch points along these 3 segments, but there's a branch point here. So we draw another line down to time t, and we color these 3 segments red. And then maybe next time we find no branch points along the black line. And so at that point, we've constructed the entire Yule tree, and then we stop. One thing to notice is that at each step in this construction, we have probability e to the minus RT of finding no branch point along the black line, because we're always encountering branch points at rate r. And so the probability that the population size at time t is equal to k, which is the same as the probability that there are exactly k steps in this construction, is the probability that we get k minus 1 tails followed by a head if we toss independent coins that come up heads with probability e to the minus r times t. So in particular, this gives a nice conceptual proof of the standard undergraduate fact that the population size at time t has the geometric distribution. Now, what this construction really does is it shows how to construct a Yule tree from what's known as a coalescent point process. So the coalescent point process goes back to the work of Leah Popovich in 2004 and to Aldous and Popovich in 2005. And the idea behind the coalescent point process is that it allows us to construct a genealogical tree from a sequence of random variables hi. So here we're going to let h1, h2, and so on be IID random variables having the exponential distribution with rate r. And we want to think of hi as representing how far back we had to look to find a branch point in the ith step of the construction on the previous slide. So we then start by drawing a vertical line on the left of height t, and then we draw vertical lines of heights h1, h2, and so on, each time drawing a dashed line to the left and stopping when we hit a vertical line. So in the picture, we start with this segment of time t, then we draw a line of height h1, which coalesces with the initial segment here. We draw a line of height h2, which coalesces with the previous segment here. We draw a line of height h3, which coalesces at this point, and so on. And then we continue this process until one of the random variables hi is greater than t, and then we stop. And the tree that we have in the end is exactly a Yule tree run for time t, in which each individual gives birth at rate r. Now, this construction can be extended to the case of branching processes in which there can be births and deaths. In that case, we construct the so-called reduced tree. That is, we condition on the event that the population survives until at least time t, and we only construct the individuals that have descendants alive in the population at time t. Now, the branching process needs to satisfy two conditions. First, all individuals in the population at time t have to be independently giving birth to exactly one offspring at some rate lambda of t. And then any individual that's born at time t has some probability q of t of having a descendant alive in the population at time capital T. So notice this second condition allows for the possibility that the death rate could be age dependent. And then Lambert and Stadler showed that one can construct the reduced tree by using a coalescent point process. And what we do is to use exactly the same construction on the previous slide, but we modify the distribution of the hi's so that the probability that hi is greater than t is given by this expression here. And the idea is that as we are going back along the black line looking for branch points, the chance of finding a branch point around time s is proportional to the birth rate lambda of s multiplied by the probability q of s that an individual born at time s will have a descendant alive at time capital T. And so the chance that we have to look back more than little t time units to find a branch point is given by the exponential of the negative of this integral. Notice that with this construction, it's easy to condition on the event that the population size at time t is equal to n. We simply replace the original random variables hi by these new random variables hi star, whose distribution is the conditional distribution of the hi's given that hi is less than t, and then we stop the construction after n-1 steps. So that just conditions the first n-1 random variables to be less than t, and the nth random variable to be bigger than t. And if we do condition on the population size being equal to n, the n-1 coalescence times are IID random variables, independent and identically distributed. Now, what we're most interested in is not a sample from the is not the genealogy of the entire population, but rather the genealogy of a sample from the population. So let's suppose we sample the individuals I and j, but not the individuals in between. Then I claim that in the coalescent point process, we need to replace the random variable hj by the maximum of hi plus 1 through hj. So let's look at this in the picture. Here we've sampled the individuals 1 and 4, but not the individuals 2 and 3. And you can see that lineage 4 gets traced back to this point and coalesces with the first lineage at the time h3, which is indeed the largest of h2, h3, and H4. Now, if each individual is independently sampled with some probability y, then the number of random variables over which we're taking the maximum has a geometric distribution with parameter y. And so we can construct the genealogical tree of the sample by using a coalescent point process. We simply have to replace the original random variables hi with these new random variables hiy, whose distribution is the same as the distribution of the maximum of a geometrically distributed number of the original random variables. This idea goes back to Tanya Stadler in 2009 and was presented in greater generality by Lambert and Stadler. But what we're most interested in is taking a sample of fixed size n. That turns out to be much harder, but there was a recent breakthrough by Amaury Lambert. Lambert showed that the genealogical tree of a sample of size n from a birth and death process at time t, conditioned on the event that the size of the population at time t is at least n, we can construct this genealogical tree, the genealogical tree of this sample of size n, exactly by using a coalescent point process. Now, to obtain a genealogical tree from a coalescent point process, we just have to explain how we get the random variables h sub I. And we use a two-step procedure. First, we choose a random variable y from the unit interval that has this probability density function here. And then, conditional on the value of y, we sample each individual independently with probability y and condition on getting a sample of size n. And so that entails choosing the random variables h1 through hn minus 1 to have this probability density function here on the interval from 0 to t. And that complicated formula simply comes from starting with the formula that you would use for the original birth and death process, taking the maximum of a geometrically distributed number of those random variables, and then conditioning that maximum to be less than t. And so you can see that this scheme is potentially quite useful for simulations. We can efficiently simulate the exact genealogy of a sample from a birth and death process without having to simulate first the entire population. And I also want to mention that although we're going to use this construction of Lambert to obtain our results, Simon Harris, Samuel Johnston, and Matt Roberts obtained the exact same formula for the joint distribution of the coalescence times by using a completely different method. And in spite of the publication dates, my understanding is that the work of Harris, Johnston, and Roberts actually appeared a few months earlier and was the motivation for Lambert's construction. Now, those formulas on the previous slide got pretty complicated. However, things simplify quite nicely if we take a limit first as the sampling time t goes to infinity, so that we're taking a sample after a large time, and then we take a limit as the sample size goes to infinity. In that case, under the appropriate scaling, we get the following much simpler coalescent point process in the limit. So we let w have an exponential distribution with rate 1. We let u1 through un minus 1 be IID random variables having the logistic distribution, which is a symmetric distribution on the real line having this probability density function here. And then we let hi be t minus 1 over r times the log of 1 over w plus the log of n plus ui. So to understand a little bit better where this formula comes from, recall that we expect the population size at time t to be approximately w times e to the rt, where w has an exponential distribution. And therefore, the time that it takes for the population to reach size n is approximately 1 over r times the log of 1 over w plus the log of n. Now, if we take a sample of size n from the population after a large time and follow the ancestral lines backwards in time, then we would expect the ancestral lines to coalesce around the time when the population size is of the order n. And that's why these coalescence times are fairly close to t minus this expression here. So in particular, this random variable W, this exponential random variable W that appears in the formula, is the same W that gives the exponential limit law for the population size. Now, I have not seen this limiting coalescent point process elsewhere in the literature. So this is actually the first thing that I'm presenting in this talk that I think is new. However, I do want to mention that Ignatieva, Hein, and Jenkins used a different method involving a random time change to show that the coalescence times are well approximated by IID logistic random variables. And so the appearance of the logistic distribution in the limit here was not a surprise. So once we have a coalescent point process construction of the genealogy, we can read off information about the internal and external branch lengths. So here's our picture of the coalescent point process with the external branches in blue and the internal branches in red. And I claim that if I is between 1 and n minus 2, so I'm setting aside the first branch and the last branch, which need to be considered separately, then the length of the internal portion of the ith branch is the positive part of hi minus hi plus 1, and the length of the external portion of the ith branch is the minimum of hi and hi plus 1. So to see that in the picture, let's consider the branch labeled 3. The internal portion, rather, is this red portion, which has length h3 minus h4. And the external portion has length h4, which is the minimum of h3 and h4. If we look instead at the branch labeled 4, there is no internal portion because h5 is greater than h4, so this positive part is 0. And the external portion is again h4, which is the minimum of h4 and h5. And of course, to obtain the total internal branch length, we can sum up the red portions of the individual branches. And to obtain the total external branch length, we can sum up the blue portions of the individual branches. Now, we can also recover information about the site frequency spectrum from the coalescent point process. This, again, is not a new idea. It goes back to this 2009 paper of Emery Lambert, who showed how to compute the exact expected value of the site frequency spectrum for a population whose genealogy can be described by a coalescent point process. Later, Champagnat and Lambert obtained some law of large numbers type results, and Champagnat and Henri computed some higher moments. Delaporte, Achaz, and Lambert considered a sample from a population, but they assumed that each individual was independently sampled with probability y, and so they got formulas depending on y. And then I also want to mention this paper of Din, Jaksic, Kimmel, Lambert, and Tabaret, who applied some of these ideas about the site frequency spectrum to cancer data. Now, the formula that's going to be important for us is this one here. And what this says is that the portion of the ith branch that supports k leaves is given by the minimum of hi and hi plus k minus the maximum of the h's in between, and then we take the positive part. So for example, in the picture, this little red branch here supports 4 leaves, 1, 2, 3, and 4. And its length is h1 minus h3. And notice that h1 is the minimum of h1 and h5, and h3 is the maximum of h2, h3, and h4. Likewise, this red branch here supports 3 leaves, 5, 6, and 7, and its length is h8, which is the smaller of h5 and h8, minus h6, which is the larger of h6 and h7. And of course, as before, we can sum up these quantities over I to obtain the total length of all branches that support k leaves. So let's now start to consider some asymptotic results for the total internal branch length. Recall that we have this approximate formula for hi when t and n are large. And we said that the internal portion of the ith branch has length, the positive part of hi minus hi plus 1, which is the same as 1 over r times the positive part of ui plus 1 minus ui. But because ui and ui plus 1 are just independent logistic random variables, we can calculate this expectation exactly. And it turns out to be equal to 1. And so when we sum this up over I, we get that the expected value of the total internal branch length is approximately n over r. And this is consistent with Duret's formula that we saw much earlier in the talk. Because recall that Duret showed that the length of the portion of the length of the branches that support k leaves should be approximately n over r times 1 over k times k minus 1. But when we sum this over k from 2 to infinity, the sum telescopes n equals 1, so we just get n over r, which is the same as our estimate of the total internal branch length. But now we can go a step further and try to estimate the variance of the total internal branch length. So for that, we need to compute the variance of the sum of these random variables. But notice that if I and j differ by more than 1, then LIN and LJN are independent. So we just need to compute the variances of these random variables and the covariances between consecutive random variables. Those are calculations involving 2 or 3 logistic random variables, which is manageable. And so in the end, we're able to calculate that the variance of the total internal branch length is approximately n over r squared. Now, to understand not just the mean and variance, but the asymptotic distribution of the total internal branch length, we need to invoke the most famous result in probability theory, which is the central limit theorem. So if we have a sequence of IID random variables with finite non-zero variance, and if we let xn be the sum of these random variables, then the central limit theorem tells us that the distribution of this sum is asymptotically normal, in the sense that if we take Sn, subtract its mean, divide by the square root of its variance, this converges in distribution to a standard normal distribution. Now, what we're going to use is actually an extension of this result, known as the central limit theorem for m-dependent sequences. And this result says that even if the random variables xi are not independent, the conclusion of the central limit theorem still holds as long as the xi's are m-dependent for some m. And so that means that for all n, the random variables x1 through xn are independent of everything that comes after xn plus m. So basically, we're only allowed to have local dependencies among the xi's. Now, recall that this random variable lin depends only on ui and ui plus 1, and the u's are independent logistic random variables. So these random variables lin are not independent, but they are one-dependent. And so their sum, which is the total internal branch length, should have asymptotically a normal distribution by the central limit theorem for one-dependent sequences. And so this leads us to the following limit theorem for the total internal branch length. So to get a limit theorem, we need to consider a sequence of processes indexed by n. So I'm going to write lambda n and mu n for the birth and death rates, Rn for the difference between the birth and death rates, and Tn for the time at which we take the sample. It's fine if you want to think of lambda, mu, and R as just being constants, but the sampling time Tn does need to tend to infinity at a certain rate along with the sample size n. So we need to make this assumption here. Recall that the expected population size at time Tn is e to the Rn times Tn. So this assumption is just saying that the sample size is much smaller than the population size. And under that condition, we get a law of large numbers. Rn over n times the total internal branch length converges in probability to 1. So basically, this is saying that the total internal branch length with very high probability is very close to n over Rn. If we make this slightly stronger assumption here, then we get a central limit theorem. We take the total internal branch length, subtract our estimate of the mean, divide by the square root of our estimate of the variance, and this quantity converges in distribution to a standard normal distribution. Now, just to make the link with Kit Curtius's talk on Wednesday, because the total internal branch length is approximately n over Rn, we can estimate the growth rate Rn of the branching process by taking n, the sample size n, and dividing it by the total internal branch length. And then this asymptotic normality allows us easily to get a 95% confidence interval for the growth rate. And that's how the statistical inference was done in the talk by Kit Curtius on Wednesday. One can also get a result for the external lengths. Recall again, we have this, this approximate formula for HI, and we said that the portion of the external portion of the ith branch is given by the minimum of HI and HI plus 1. Because of the minus sign here, we really need to consider the expected value of the maximum of UI and UI plus 1. And by a short calculation involving the logistic distribution, this turns out to be equal to 1. Now, because the coalescent tree is star-shaped, each external branch has a length approximately equal to the sampling time t sub n. And so we would expect the total external branch length to be very close to the sample size n times the time t sub n. And therefore, we would expect Rn over n times the total external branch length to be approximately Rn times Tn. There's a correction of log n coming from this term, and there's a correction of 1 coming from the UIs. But then these quantities here will converge in distribution as n goes to infinity to the logarithm of an exponential random variable. So in particular, the dominant source of fluctuation in the total external branch length is coming from the exponential random variable w that governs the long-run population size. And we also do have a joint convergence result for the internal and external branch lengths, and they turn out to be asymptotically independent. So let's turn now to the site frequency spectrum. Recall that the portion of the ith branch that supports k leaves is given by the minimum of HI and HI plus k minus the maximum of the h's in between. Translating this into the u's, this is 1 over r times the minimum of k minus 1 logistic random variables minus the maximum of 2 logistic random variables. We can calculate the expectation of this by integrating over x the probability that ui and ui plus k are less than x, and the k minus 1 random variables in between are greater than x. And because we know the joint distribution of independent logistic random variables, we can compute this integral exactly, and we get 1 over r times k minus 1, which of course agrees with Duret's formula. We can also compute covariances between these quantities for different values of I and k, although that's a much more complicated computation. And we can deduce the asymptotic normality of the total length of all branches that support k leaves from the central limit theorem for k-dependent sequences, much as we did for the total internal branch length. And so that finally leads us to our main limit theorem for the site frequency spectrum. If we assume that the sample size is much smaller than the population size, then we have a law of large numbers. Rn over n times the total length of all branches that support k leaves converges in probability to 1 over k times k minus 1. Under this stronger assumption, we get a central limit theorem. We let mu k be n over r times k minus 1. That's our estimate of the expected value of LKN. And then we consider this random vector, where we start with LKN, subtract our estimate of the mean, and normalize by multiplying by Rn over the square root of n. And then the distribution of these random vectors converges as n goes to infinity to a centered multivariate normal distribution. We do have an explicit formula for all of the covariances, but it's a very complicated expression, and I've chosen not to try to write it down here. Now, before we move on, I want to mention briefly 2 extensions of this result. So first, we have focused entirely on the case of supercritical birth and death processes, where the birth rate is greater than the death rate. That's the case that's most relevant for cancer applications. But all of the theory that I outlined still basically applies in the case when we have a critical birth and death process and the birth rate is equal to the death rate. And the results that we get in that case are pretty much the same as the asymptotic results that one gets for Kingman's coalescent. Also, we have focused almost entirely on studying the branch lengths, and we've just assumed that mutations happen at some constant rate along the branches. But in some applications, it may be more natural to assume that mutations arise only at the times when a birth event occurs. My graduate student, Yubo Shuai, worked on this problem, and he showed that in the setting when mutations occur only at the times of the birth events, there is still an efficient algorithm for simulating the exact genealogy of a sample from the population, this time with marks along the branches of the tree to indicate the times of mutations. Also, the asymptotic results that one gets for the site frequency spectrum are almost exactly the same once you do the right scaling, almost the same as what you get in the case when the mutations arise at a constant rate along the branches. So I now want to move into the last part of the talk on populations with spatial structure. So probably one of the reasons why we got such good results for blood cancer data by using a model of an exponentially growing population is that blood cancer cells can move around rather freely. However, for solid tumors, one would not expect them to be able to grow exponentially because their growth is constrained in space. So we would also like to be able to consider a population model that accounts for these spatial constraints. So one famous model, which goes back to 1961, is known as the Eden model. So in the Eden model, cells are represented by particles at sites on the two-dimensional integer lattice. We begin with one particle at the origin, and at each time step, we add a new particle at a site that is chosen at random from the sites that are vacant but are adjacent to an occupied site. So in this picture, if the black dots represent the locations of the particles at a certain time, then a particle could be born at this red site in the next time step. If we want to keep track of the genealogy, we can simply choose a neighbor at random to be the parent of the new individual. So in the picture, either this particle or this particle would be designated as the parent of the red particle. Now, the Eden model is one of a large class of random growth models that is conjectured to belong to the so-called KPZ universality class. The KPZ equation, which is named after Carter, Parisi, and Zhang, is a stochastic partial differential equation which models the growth of a random interface. Here, h of t comma x represents the height of the interface at the time t and at the spatial location x. Now, I'm not going to discuss this stochastic partial differential equation in detail. It's very challenging even to understand what this equation means because the equation in this form is not well posed. However, from scaling arguments, one can determine that if the growth of the interface is governed by the KPZ equation, then after a large time t, the fluctuations in the height of the interface at a particular location are on the scale of t to the 1/3. And if we consider the height of the interface at two locations that are separated in space by at most order t to the 2/3, then one will see correlations between the fluctuations at those 2 points. So the KPZ equation is believed to describe random interface growth under a very wide range of circumstances. To give just one example, let's say you take a sheet of paper and light the bottom of it on fire. Then the random curve that separates the portion of the paper that has burned by time t from the portion of the paper that has not yet burned should be well approximated by the KPZ equation. And likewise, the KPZ equation should approximately describe the random curve that follows the outer boundary of the occupied region in the Eden model. So the study of the KPZ equation has been one of the most active areas of research in probability theory over the last decade or so. And just within the last few years, a mathematical object called the directed landscape has been constructed. And this directed landscape is conjectured to describe the scaling limit in all models in the KPZ universality class, although up to this point, the convergence has only been proved rigorously in one specific model known as last passage percolation. So I'm going to just briefly introduce the directed landscape, which was constructed by Dauvergne, Ortmann, and Virag in 2022. Both Duncan Dauvergne and Balint Virag are at the University of Toronto. So Toronto is sort of the birthplace of the directed landscape. So we are going to let R4 with an arrow be the set of all pairs of points x comma s and y comma t in R2, where s is less than t. And then the directed landscape is a random function from this space to the reals. Roughly speaking, we think of L of x, s, y, t as assigning a distance between the points x comma s and y comma t. Although because of the scaling that's used, this quantity could be negative, and because of a sign convention, it satisfies the reverse triangle inequality rather than the usual one. In our setting, we can roughly think of this quantity as representing the time that it would take for a tumor to spread from y comma t to x comma s. Now, the most important property of the directed landscape is this scaling property, which says that if we scale the horizontal coordinate by rho squared and scale the vertical coordinate by rho cubed, then everything looks the same. These two quantities on the two sides here have the same distribution. Now, because the directed landscape gives us a notion of distance between points, we also have a notion of geodesics representing the optimal path between 2 points. And therefore, if we have a point x comma s in R2, we can define the so-called infinite upward geodesic starting from the point x comma s to be a continuous function g such that g of s is equal to x. The restriction of g to a finite interval is a finite geodesic, and g of t over t goes to 0 as t goes to infinity. This is just saying that asymptotically the geodesic points in the upward direction. And one can show that in the directed landscape, with probability 1, there are infinite upward geodesics starting from every point. And if we take any particular point x comma s, with probability 1, the infinite upward geodesic is unique. Now, in the setting of a population model, we can think of the points along this infinite upward geodesic as representing the ancestors of the individual in the population located at x comma s. So let's say we have a population that evolved according to the Eden model. And then after a large time, the population will occupy roughly this circular region. Now, the directed landscape will not capture the circular geometry of the Eden model, but we can use the directed landscape to approximate the genealogy of a sample of individuals taken from a patch that's far away from the center of the circle. So that's illustrated in the picture on the left. Here are 5 individuals that we've sampled from the population. And when we trace back their ancestral lines, they all eventually go back to near the center of the circle. So to approximate this genealogy using the directed landscape, what we can do is to take a box of area n. We sample lowercase n points uniformly at random from this box, and then we consider the infinite upward geodesics starting from these points. And these geodesics will eventually coalesce, and then we can let LKN denote the sum of the vertical lengths of all of the branches along these geodesics that belong to k of the n geodesics. So in the picture, these two red branches are each ancestral to 2 of the 4 sampled points. And so L2n would consist of the vertical lengths of these 2 red branches. And so what we're going to do is to ignore the very technical and challenging question of proving convergence of the discrete model to the directed landscape. And we're just going to focus on studying these LKNs for the directed landscape. And here is the result that we get. It's perhaps a bit premature to call this a theorem because we are still finalizing a few of the details of the proof, but we are confident that everything works out. And so the theorem says that for all positive integers k, LKN divided by n converges in probability to a constant times gamma of k minus 2/5 divided by k factorial as the sample size n goes to infinity. Notice that gamma of k minus 2/5 over k factorial is asymptotically k to the minus 7/5 as k goes to infinity. So just as we got k to the minus 1 for Kingman's coalescent and k to the minus 2 for exponentially growing populations, we get k to the minus 7/5 for populations in the KPZ universality class. This k to the minus 7/5 exponent was predicted using non-rigorous methods in this earlier physics paper by Fusco, Gralka, Kaiser, Anderson, and Halaschek. Now, one thing to note here is that in this theorem, we fix k and we let the sample size n go to infinity. We do expect that this result would give fairly accurate results in the Eden model as long as k is much less than n to the 5/6 power. For larger values of k, the points that are ancestral to k of the sampled points would be close to the center of the circle. And there, the circular geometry would play a role, and the directed landscape approximation is no longer valid. At this point, we do not have rigorous results in that regime. However, non-rigorous calculations in this physics paper suggest that when k is larger than n to the 5/6, but smaller than n, the site frequency spectrum starts to decay like k to the minus 5. So I want to explain briefly the idea behind this k to the minus 7/5 behavior. So let's suppose we consider 2 infinite upward geodesics that begin 1 unit apart. Well, then they are going to coalesce after a time that's of order 1. Now, recall that in the scaling of the directed landscape, if we scale the horizontal axis by r, then we need to scale the vertical axis by r to the 3/2. And so 2 infinite upward geodesics that begin r units apart will coalesce after a time that's of order r to the 3/2. So in this picture, if we consider the infinite upward geodesics starting from points in one of the red ovals, they all go through the black dot at the top of the oval. And so the black dots here are the points along this top line that have descendants surviving r to the 3/2 time units into the future. And because these ovals have a width of order r, these points along the top line are spaced order r units apart, and the region representing the descendants of these individuals has an area of order r to the 5/2. And so letting k be r to the 5/2, we see that the points along the top line that have order k descendants should be spaced order k to the 2/5 units apart. And that suggests that the points along the top line that have exactly k descendants should be spaced order k to the 7/5 units apart. And so along any horizontal line segment, the expected number of points that are ancestors of exactly k of the sampled points should be proportional to k to the minus 7/5, and that's what leads to our result. Now, in some applications, it may be more natural to assume that we have a sample of individuals taken not from the entire population, but from the outer boundary of the population. That's illustrated here in the picture on the left. Here are 5 individuals that we've sampled from the outer boundary, and we can trace their ancestral lines back to the center of the circle. To approximate this genealogy using the directed landscape, we can choose n points uniformly at random from the portion of the x-axis between 0 and n. We can consider the infinite upward geodesics starting from these points, and we can define LKN as before. And in this case, we get that for all positive integers k, if we take LKN divided by n, this converges in probability to a constant times gamma of k plus 1/2 over k factorial as the sample size tends to infinity. This quantity on the right-hand side decays like k to the minus 1/2 as k gets large. And once again, this k to the minus 1/2 behavior, which is quite different from what we saw when sampling from the entire population, was predicted in the physics literature in this 2022 paper by Egdami, Paulos, and Fusco. As before, in this theorem, we fix k and let n go to infinity, but we would expect the results to be approximately valid in the Eden model as long as k is much less than n to the 2/3. This condition ensures that points that are ancestors of k of the sampled points are going to be much closer to the boundary than to the center of the circle. Now, we have not yet tried to apply these results to cancer data, but I do believe that the results may be relevant for the study of some biological populations. In this 2012 paper, it was shown that data on the growth of cervical cancer agrees with some of the predictions of models in the KPZ universality class. And KPZ type growth has also been observed in bacterial populations and in some invasive plant species, such as white clover. I do think there are some limitations in trying to apply these results to cancer data. First of all, many solid tumors are 3-dimensional, whereas these results apply only to 2-dimensional growth processes. Secondly, in the Eden model, all of the growth occurs along the boundary of the tumor. And so these models do not account for the possibility that we may have births and deaths in the interior of the tumor. Also, some research suggests that, that the boundary of many tumors is more smooth than what would be predicted by models in the KPZ universality class, perhaps because some of the cells near the boundary of the tumor can move around more than what these models predict. Nevertheless, I do hope that these results will be a positive step towards understanding the mutational patterns in spatially structured populations. And I'll stop there. Thank you.